Hello everybody, in this video lecture we will cover the cell cycle and cancer. So this is a PowerPoint that I will use. Let's go ahead and begin. Cell's complete complement of DNA is called its genome. The genome comprises of chromosomes. Each eukaryotic chromosome contains one very long DNA molecule, typically bearing thousands of genes. The number of chromosomes in eukaryotic cells depend on the species. On this picture below, you can see on the bottom that represent DNA molecule. Not entire DNA molecule is made of genes. Genes are just part of DNA that code for protein synthesis. So here's one gene, then we have intergenic region, and then another gene. That means this part of DNA codes for a protein. This part of DNA codes for a protein. This area in between, it can be regulatory area. Maybe it regulates how much of this protein we need, or do we even need to synthesize it, right? So that would be some region that does not code for proteins. So DNA made of genes and intergenic regions. Now this DNA make chromosomes, right? And we will look at the next slide. We will look into more details of that. Now, all the chromosomes make a genome of a particular organism. On this bar graph, I would say, you see a number of chromosomes for different species. So a human, we have 46 chromosomes, but it doesn't mean we have more than all other animals or less, right? It's just our number. If you look at your dog, for example, they have 78 chromosome. This rat has 102, koala has 16. But if you notice, all of these numbers are even numbers. And this is because those animals are sexually reproducing animals. So all of these examples of animals here, they reproduce sexually. That means to reproduce, it requires two parents, right? So for human, one person cannot reproduce. We need two parents, and each parent will donate half of this number. So humans, we have 23 pairs. So you have 23 pairs. So you, when you make eggs or sperm, this egg and sperm will carry only half of this DNA, 23 chromosomes, right? So that's why all these numbers are even. Chromosomes are made of chromatin. Chromatin is not visible in a cell until cell division occurs. So if you look at picture over here, that's one cell. Here's the second cell. Those are plant cells, but the same is true for animal cells. So if you look here inside this nucleus, you don't see chromosomes. Chromosomes are very distinct structure, can be seen in this cell. Well, then what is inside? Inside is chromatin. And when chromatin condenses, then we can see chromosomes. So this condensation of chromatin happens only when cell is dividing. So chromatin is a complex of macromolecules found in cells consisting of DNA, proteins, and RNA. Proteins have special name, they're named histones. The primary function of chromatin are to package DNA into a more compact, denser shape to prevent DNA damage. So on this diagram, you see a nucleus. This purple color represents nuclear envelope, nuclear pores, and what is inside the nucleus is chromatin. So it's pretty loose, structure, like a spaghetti inside a bowl, right? But if you look at this chromatin, you can see that it's made of DNA, right? So that's a DNA helix, and those are proteins called the histones. So DNA is wrapped around these histones, making, this highly organized structure, but it's still not very condensed, right? When chromatin condenses, this is how it looks. Then we call it chromosome, right? Chromosome has arms, right? So here's one arm, so those are arms. In the middle, we have centromere. Telomeres are at the end. And this chromosome here look like X because this is already duplicated chromosome. And each part of this duplicated chromosome is called sister chromatid. So this is one sister chromatid, this is a second sister chromatid, and they will be held together in the centromere until cell divides, and these two sister chromatids will be pulled apart, and each one will be a complete, functional chromosome after cell division. Human body cells, somatic cells, have 46 chromosomes, configuration known as diploid or 2N. So cells of your body are somatic cells, they are diploid, 2N, and for human we have 46 chromosomes. Human cells that contain one set of 23 chromosomes are called gametes or sex cells. These eggs and sperm are designated as 1N or haploid. So if you look at this picture, you see that diploid organism, you not only have 46 chromosomes, you have actually 23 pairs. This pink chromosome and this pink chromosome make one pair. 2 chromosomes, 1 pair. Those chromosomes are also another pair. Here's another pair. So here we have 3 pairs of chromosomes or 6 chromosomes. A haploid organism has 1 chromosome from each pair, right? So DNA is not lost, really. The amount of genes are, all genes are present, right? But you just have 1 chromosome from each pair. Those chromosomes that make a pair, they called homologous chromosomes. They're not identical, but they're not completely different. They are homologous. So you get 1 pink from mom, this pink from dad. And again, when I say pink or purple or yellow, it's not how your chromosome look like, right? It's just representation. But this chromosome maybe you get from mom, this from dad, this from mom, this from dad, right? So those are homologous chromosomes. So this pink and this pink are homologous. Pink and purple are not homologous. Homologous chromosomes have the same shape, same size, the same structure, and they carry the same genes, right? What can be different? Can be different version of these genes, can be different alleles, but genes are the same, right? So you and me and any of our classmates, we all are diploid organisms. So if you look inside our cells, your skin cell, your liver cells, you will find 23 pairs of homologous chromosomes, right? So here's a female, her cells are diploid. This is male, his cells are diploid. But when we produce sex cells, ova or sperm, those cells carry only 1 set of homologous chromosomes, 23 and 23. But when they fuse together through fertilization and zygote is formed, that's the first cell that will grow into new organism. This zygote will have 46 chromosomes in 23 pairs. And then this cell will divide until we have embryo, right, and then newborn, and then we grow and so on, became adults. So we all started from one single cell that have half of the chromosome from mom, half from dad, making it 46. Karyotype is an image that reveals an orderly arrangement of chromosomes. So this is karyotype of an individual. And if you look here, it has X and Y chromosomes. That means this is karyotype of a male. Homologous chromosomes are matching pairs of chromosomes that can possess different versions of the same genes. Right, so this is chromosome number 1, and this is chromosome number 1 prime, and those are homologous chromosomes. Number 2, homologous to number 2 prime. So they have the same genes, but they might carry different versions of the same genes. Humans have 2 different sex chromosomes, X and Y, and 22 pairs of matching chromosomes called autosomes. So if you are a female, then you have XX chromosomes as your sex chromosomes. If you are a male, you have XY chromosomes that are sex chromosomes. So X and Y are not homologous, right? Only in in guys, we have 1 pair of chromosomes that are not homologous. The cell cycle is an ordered series of events involving cell growth, cell division that produces 2 new daughter cells. Cells on the path to cell division proceed through a series of precisely timed and carefully regulated stages of growth. So cell cycle represented by pie diagram, and what's shown here in red color, M, this is where cell dividing, and it's about, I don't know, like maybe less than 20% of this cycle. Everything else is called interphase, and this is where cell is growing, then cell is replicating a DNA, continue to grow, prepares for division. In an M phase, cell divides, right? So here's our newly born cell. We actually have 2. Each of them will have its own cell cycle. So this cell will grow, then the cell will make copy of all its chromosomes. So you see, that's our chromatin. Now we have 2 copies. Now cells continue to grow and performing all its function, preparing for division, and then it's divide. The cell cycle has 2 main phases, interphase and the mitotic phase. So here's everything shown in gray color is interphase, and mitotic phase is this part of the cell cycle. During interphase, the cell grows and DNA is replicated. During the mitotic phase, the replicated DNA and cytoplasmic contents are separated and cell divides. Now, cell only divides during M phase, right? All the preparation for division, including DNA synthesis or replication, happens during interphase. During interphase, the cell undergoes normal processes while also preparing for cell division. The 3 stages of interphase are called G1, S, and G2. And G stands for growth, so growth phase 1. S stands for synthesis, and G2, again, cell growth. In the S phase, or synthetic phase, DNA replication results in the formation of 2 identical copies of each chromosome, sister chromatids. They are firmly attached at the centromere region. So here's 2 chromosomes over here. They are homologous chromosomes. So they make a pair. So that means there's the same shape, same size, centromere located in the same area, and they carry these same genes, right? But one you get from your one parent and one from another parent. one from mom, one from dad, right? So those are homologous chromosomes, and they are not replicated. But during replication, the identical copy is made, right? So identical copy of this green chromosome and identical copy of the purple chromosome. So now we still have two homologous chromosomes, but each one is replicated and each one is made from two sister chromatids. So these homologous chromosomes, we have two of them, but we have 1, 2, 3, 4 sister chromatids. Now, sister chromatids are attached to each other in this centromere region. So here's our cell cycle again, but here we're gonna look at one more phase. It's called G0 phase. G0 phase is a phase in a cell cycle in which the cell is neither dividing nor preparing for division. Hence, it's a resting phase. Some cells enter G0 temporarily until external signals triggers the onset of G1. Other cells that never or rarely divide, such as mature cardiac muscles, nerve cells, remain in G0 permanently. Right? So not every cell in your body will divide, and if cells don't divide, they just enter this G0 phase, resting phase. Okay, so again, this is our interphase. Here's our G0 phase or cell cycle arrest. And here we will talk about M phase. The M phase or mitotic phase includes two overlapping processes. Mitosis, in which the nucleus and its contents divide evenly into two daughter nuclei, and cytokinesis, in which the cytoplasm is divided into two. So here's mitosis, where nuclear division occurs, and cytokinesis, cytoplasmic division occurs. To ensure the proper division of the cell, there are control mechanisms known as cycle checkpoints. Each step of the cell cycle is monitored by internal control called checkpoints. There are three major checkpoints in the cell cycle: G1, G2, and M. So if you look over here, that show actually here's the G1 at the end of G1 phase. Here's G2 at the end of G2 phase, and one in the M phase, so it's M checkpoint. So integrity of the DNA is assessed at the G1 checkpoint. Proper chromosome duplication is assessed at G2 checkpoint. And attachment of the kinetochore to a spindle fiber is assessed at the M checkpoint. So what is checked in G1? In G1, just checked is, DNA inside the cell functioning normal. If cell is growing correctly and cell performing all its function. And if cell is growing correctly and it's performing all its function, that means that DNA does not have much errors, no mutations, right? So G1 is fine, DNA is normal. If DNA is working normally, then cell can synthesize this DNA, make a copy of a DNA, right? It's just allow it to make a copy of DNA. If DNA already has so many mutations and cell is not functioning, there is no reason to duplicate this DNA that is not functioning anymore, right? So after DNA is replicated in G2 checkpoint, what is assessed? Assess this replication. Was DNA synthesis done properly? Is DNA replicated correctly? Because if cell make mistake, then this checkpoint will prevent the cell from division. But if DNA synthesis was fine, DNA was replicated, then cell will proceed to division. Now, M, it's right in the metaphase, right? So when cell is ready to separate its sister chromatids, So that's what this checkpoint gonna assess. What it says here, if each kinetochore attached correctly, we didn't cover the mitosis yet. So just now remember that M checkpoint is checking if everything is going right during this division process, right? So now what is cancer? Cancer is collective name for many different diseases caused by a common mechanism, uncontrolled cell division. Cancer is a disease of the cell cycle. Cancer cells do not respond normally to the cell cycle control system. So we just talk about these checkpoints. So now what's gonna happen, I will go back for a second. What's gonna happen, for example, over here, if this cell has a DNA mutation and it's not functioning properly, what's gonna happen at this point of time? Then this cell will destroy itself. Cell will go through the process called apoptosis. So this cell pretty much will die. It will never synthesize DNA and it will never divide, because you have this mechanism built into your cells that prevent cells that are damaged from division, right? So if your cell's fine, it's gonna synthesize DNA. But what if during DNA synthesis, mistake is made? What if it's like really bad mutation? Then at G2 checkpoint, cell will go through apoptosis. So cell will die. It's a programmed cell death. That's good for living organisms, because we don't want our cells that are not functioning normally continue dividing. Now, cancer cells, they skip those checkpoints. They do not respond to this checkpoint system. Cancer cells that then can form tumors. Tumors are abnormally growing masses of body cells. If the abnormal cells remain at the original site, the lump is called benign tumor. So here, for example, you have some duct, maybe, small intestine, and those are cells. And some cells start dividing out of control, so they don't respond to this control system. So they form a mass of cells called tumor. But this is benign tumor because it stays at the point of origin. But some tumors start moving through the body. It's invading other tissues. This is called metastasis, and this tumor is called malignant tumor. So the spread of cancer cell beyond the original site of origin is metastasis. Malignant tumors can spread to other parts of the body and interrupt normal body functions. A person with malignant tumor is said to have cancer. So over here you also see the tumor growing, that's a breast cancer. And it starts invading the neighboring tissue, and it even can travel through blood or lymph system to other organs, right? So that's a metastasis already of the cancer. All cancers begin with a gene mutation that gives rise to a faulty protein that participates in the process of cell reproduction. Remember, cancer is a disease of cell cycle. So cells start dividing without control. Because of the mutation, the plates of the cell cycle speed up, because of the mutations, the pace of the cell cycle speeds up as the effectiveness of the control and repair mechanism decreases. Uncontrolled growth of the mutated cell may result in tumor. So all cells start with a gene mutation. So here's a cell, nucleus inside, and DNA within this nucleus. So this cell does not have any mutated DNA. Now, if mutation happen, and if mutation happen in a specific gene, right, that's responsible for cell division, then this cell can start or may start divide without control and form a tumor. Now, what genes? Because it it can be just any gene. It need to be, or they are need to be specific genes where mutation happen. So here's those genes. In your normal cell, so a person without tumor, without cancer, we do have proto-oncogenes. Yeah, this sounds already scary, proto-oncogenes, but those are normal genes, right? They code for positive cell cycle regulation. So they promote cell division. Your cells need to divide. So you have this special proto-oncogenes, and this genes, it's like a, allow your cells to divide, right? Your cells represented by a car over here, right? Now, proto-oncogenes are normal genes, but when they are mutated, they become oncogenes. And oncogenes that cause a cell to become cancerous. Now, look, you always have two chromosomes. You always have a pair with the same genes. So here's a chromosome from your mom, this is chromosome from your dad, and here's the normal proto-oncogenes. What they do, they allow your cell to divide, they regulate cell division. You need cell division. Now, what happens if mutation happen in one gene? Well, a mutation happen in one gene, well, you accelerate cell division. It's like you push on a gas in the car, but you still have one normal gene. So this is not that dangerous situation yet, right? Because you still have normal gene, but you, well, not you, somebody. Okay, let's say a patient. So this patient has mutation in one proto-oncogene. So this patient has one oncogene and one normal gene, but the division accelerates towards the cancer. But fortunately, we have other genes that prevent cell from division. Those genes are called tumor suppressor genes. So this one was proto-oncogene, and they positively control cell cycles. That means they promote division. Tumor suppressor genes, they code for negative regulatory proteins. So they prevent cells from division. The type of regulations, and when activated, can prevent the cell from undergo uncontrolled division. There are several tumor suppressor genes. Here, example p53 gene. Now, this p53 gene prevents cells from uncontrolled division. So even if this person has mutation, and this mutation leads to formation of oncogene, so this person has one oncogene, this person still has tumor suppressor genes, p53, that will prevent cell from division, so no tumor. But if p53 gene is damaged, right, then it can be a problem. But what p53 gene does, if damage is detected, p53 triggers repair mechanism. If repair are unsuccessful, p53 signals apoptosis, so cells dies. A cell with an abnormal p53 protein cannot repair damaged DNA and cannot signal apoptosis. So cells with abnormal p53 can become cancerous. So now if mutation happened in p53 and the previous mutation happened in a proto-oncogene, then the chances to have cancer goes up, right? So here's the cell division. Proto-oncogenes stimulate cell division, and tumor suppressor gene inhibits cell division. So when those genes are not mutated, then, people are safe, right? Because we do need cells to divide, and we do need to inhibit unwanted division. The problem happens when mutation occurs into these genes. Now, if mutation occurs in proto-oncogenes, that stimulate cell division. And when it's mutation happen in tumor suppressor genes, there is no inhibition, right? So here it shows normal P53 gene. So let's say DNA is damaged, cell cycle is abnormal, then P53 gene will what? It will stop cell division, so cell cycle arrest. Then it will trigger DNA repair and cell cycle restart. If DNA cannot be repaired, P53 can send signal for apoptosis, programmed cell death, right? So then cell dies. Now, if mutation happen in P53 gene, right, DNA is damaged, cell cycle is abnormal, but P53 cannot do its work. Now cell cycle continues and cells can become cancerous. So here we have pretty much kind of like everything together. We have our guess, that is oncogene, and we have our break, the tumor suppressor gene. So this is when your car drive normally, right? If you need gas, right? Otherwise you cannot go anywhere. And your gas in your cells are proto-oncogenes. And you have your break, and this is tumor suppressor gene. Now, when mutation happened in a proto-oncogene and it became oncogene, and let's say half of a chromosome is lost or mutation happened in this gene, then your car speeding up really, really fast. And if mutation happened in tumor suppressor gene, this car doesn't even have a brake, right? So that's what happened when people have cancer. So here's some example of oncogene, tumor suppressor genes. We also have DNA repair genes. Let's say, so here's right examples. Here's our P53 and BRCA1, BRCA2. Those are example of genes that are responsible for DNA repair, and people that have mutation in these genes are more susceptible to breast cancer. Okay, so that was the last slide. So I'm reminding you that we cover cell cycle and briefly cancer. Thank you for watching, and I hope it was helpful.